Hi everyone and welcome to Miss Estruch Biology. This video is going to be the complete topic 2, which has been so highly requested on Instagram that yes, it is finally here. Now if you are new here, my name is Miss Estruch and I've been teaching for 14 years now, and I know just how challenging A-level biology can be and preparing for exams, and that's why I make lots of content to help you on YouTube and Instagram. Now if you do need even more help, then check out my sponsor for today's video, which is Biosnip. Now I actually follow them anyway, even though they are the sponsor, because their content is so useful for A-level biology. They will post different diagrams, graphs, content, so you can test yourself and really get familiar with the exam technique, as well as the daily quizzes that they post on their stories. So go check out Biosnip for more extra daily help. I'm also really excited to finally be able to tell you that my topics 1 to 4, which is the Year 12 AS content workbook, is now out. So if you haven't already got your copy of my active recall workbook, then head to the link in the description and get your copy to help you with your revision, consolidation, and just generally acing your A-level biology. But for now, let's get into topic 2. So within the eukaryotic cells, the key organelles that you need to know the structure and function of are all the ones listed here, and we're going to go through each of them one at a time. The nucleus, the key structures within that are the nuclear envelope, nuclear pores, nuclear plasm, chromosomes, and the nucleolus. And the overall function of the nucleus is where DNA replication occurs, and it's a site of transcription, which is where mRNA is made, and it contains the DNA, which is the genetic code for the cell. We also have within the nucleolus, that is the site of RNA production, and it's where ribosomes are made. The endoplasmic reticulum, you have the smooth and the rough. And we can see here the top diagram, we've got the rough endoplasmic reticulum, and we can tell because we've got the ribosomes on the outside. And the other side of this top diagram is the smooth endoplasmic reticulum, and that has no ribosomes on the outside. Now the RER, which is the rough endoplasmic reticulum, that is where protein synthesis occurs because of those ribosomes on the outside. The SER, the smooth endoplasmic reticulum, that is where you have the synthesis of lipids and carbohydrates, and they also get stored there as well. The Golgi apparatus is a folded membrane making this cystine shape, and we have vesicles that will pinch off from those cystinae once whatever it is that's going to be modified has been modified and packaged. And we've got the whole list here of the different things that would happen. So it could be that the carbohydrates are being added to proteins to make glycoproteins, which might be embedded within the cell membrane. You can also produce secretory enzymes, carbohydrates, transport and modify lipids. We get lysosomes. Molecules are also labelled with their destination, essentially meaning they're going to have a molecule added so they'll be able to bind to receptors on the target cell for where that molecule is needed. Lysosomes are bags of digestive enzymes, and this is often involved in phagocytosis. So the enzymes that are required to hydrolyze the bacteria or virus, whatever it might be, would be in a lysosome. There'll be exocytosis, which is where the products are going to be released to the outside after it's been destroyed. The mitochondria is the site of aerobic respiration. So you'll have lots of ATP production here. It's a double membrane organelle with the outer membrane, and the inner membrane is this folded part making up the cristae. And the inner membrane is where oxidative phosphorylation happens, which is one of the key stages in aerobic respiration. They have their own loop of DNA, which is very similar to prokaryotic DNA. And this is so that they can code for the enzymes that they need in respiration. Ribosomes, now these are found in prokaryotic and eukaryotic cells, and that is because they are not membrane bound. Ribosomes are just made up of rRNA and proteins. You do have different sizes though, so the 80S is the larger size ribosome in eukaryotic cells, 70S is the smaller sized ribosome found in prokaryotic cells, as well as in mitochondria and chloroplasts so that they can make their own enzymes. And it's the site of protein synthesis. The vacuole, which is only found in the plant cells, not in animal cells, this structure is filled with fluid and surrounded by a single membrane called a tonoplast, and this is what helps to make the cell turgid and provide support. You'll have temporary stores of sugars and amino acids within the fluid as well, and the pigments within it may colour the petals, and that's what helps to attract pollinators. Chloroplasts, again, only found in plants, and this is the site of photosynthesis. You have, again, it's a double membrane organelle, and we have the outer membrane, the inner membrane, and then on the inside we have even more membranes, and that is these thylakoid membranes which stack up to create what we call a grana if it's plural, or just a granum if we're just looking at one of those stacks. And the thylakoid membranes are embedded with proteins and pigments such as chlorophyll, and therefore it's the site of the light-dependent reactions in photosynthesis. The stroma, which is the fluid part surrounding the thylakoid membranes, you have lots of enzymes in this location which are needed for the light-independent stages of photosynthesis. Cell walls are not in animal cells, but they are in fungi and plant cells, and they help to provide structural strength to the cells and prevent them bursting if lots of water moves in by osmosis. And that's because they contain a particular molecule that provides strength, and in plants that's cellulose, and in fungi that would be chitin. The plasma membrane is found in all cells, and this is in the cell membrane, so the cell surface membrane, but also when we say it's a double bound or membrane bound organelle, it's also made up of this phospholipid bilayer with the different molecules embedded within it. And the function of the plasma membrane is it controls what can enter and exit the cell. Prokaryotic cells, for example bacteria, you need to be aware of the key differences. So it could come up as a comparison or a contrast question. So first of all, the cells are much, much smaller. They don't have any membrane-bound organelles. They do have ribosomes though, because ribosomes are not membrane-bound, but they have the 70S size ribosomes. They don't have a nucleus because that is a membrane-bound organelle. So instead, their DNA will just be as a single loop, which is free within the cytoplasm. They do have a cell wall, and that is made up of murin. Sometimes they might also contain plasmids, which are additional loops of DNA. They might have a capsule around the cell, which is to help prevent desiccation and to evade the immune system. And sometimes they'll have flagella. It might be one, it might be many, which is to help them to be able to move. But those aren't always found, those are only sometimes found in prokaryotic organisms. Viruses are acellular and non-living. The structure of virus particles consists of genetic material, capsid and attachment proteins, which we can see over here. So we have the genetic material in the middle, we have the capsid, and then we have the attachment proteins, or here they're labelled envelope proteins. Viruses replicate inside of host cells, and that is why it's very difficult to destroy them, because you would have to destroy the host cell to be able to destroy the virus. Now you need to know methods of studying cells, and the reason this is so important is the way we've discovered these internal structures or organelles is because of these advances in the methods to study cells. So advances in microscopes, and then also cell fractionation and ultracentrifugation to be able to isolate particular organelles to examine. So we'll go through each of these methods of how to study cells. First of all, the microscopes, there's a couple of key definitions you need to be aware of. You also need to know the three main types of microscope. There are optical microscopes, which are the light ones, which you'd be using in school. You then have electron microscopes, but you can have either a transmission electron microscope or a scanning electron microscope. The definition of magnification is how many times larger the image is compared to the object. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate. And the resolution of an optical microscope is determined by the wavelength of light, and the resolution of the electron microscopes is determined by the wavelength of the beam of electrons. And because that wavelength is much shorter, that is why they have a higher or a more powerful That is why they have a much higher resolution, meaning that the distance between the two objects is smaller. So let's compare the two general types of microscopes in more detail. The optical microscope, we've already said it's the light, but it is a beam of light. It's a beam of light that is condensed to create the image. In contrast, an electron microscope, it's a beam of electrons which is condensed to create the image. And what is used to condense the light and the electrons is also different. The beam of light is condensed using a lens, whereas the beam of electrons is condensed using electromagnets. The optical microscope has a poorer resolution due to the fact that light has a longer wavelength. So that means that the electron microscopes, because electrons have a shorter wavelength, they have a much higher resolving power. There's also a lower magnification for the optical, higher for the electron microscopes. You can have colour images though, and you can view living samples with the optical microscopes. So those are two big advantages. The electron microscopes can only produce black and white images, and because electrons can be easily absorbed, you have to have your sample in a vacuum. And because it's in a vacuum, you can't actually observe any living samples using an electron microscope. Now, just to go back to this black and white image point, you can still get coloured images if colours are added on programs like Photoshop afterwards. Because the optical microscopes have a lower resolution, small organelles in a cell are not visible. So you can't see details of the mitochondria, chloroplasts, ribosome. You can just see some of the large organelles, like we can see here. You can see the nucleus, you can see the DNA, you can see the chromosomes. The electron microscopes, they have much higher resolving powers, as we said, and the difference between the transmission and the electron is that the transmission, you have extremely thin specimens, and the electron gun produces a beam of electrons that transmits or passes through the specimen. So some parts of the specimen will absorb the electrons and therefore appear dark. Some parts won't, and the electrons would have passed through and they appear lighter. And we get these 2D images where we have the darker and lighter depending on whether the electrons were absorbed or not. And because there's a higher resolution, that is why we can actually see the details internally of some of those small organelles. A scanning electron microscope creates 3D images in contrast. The specimen does not need to be thin, and that's because the electrons are not going to be passing through the specimen. Instead, the electrons are beamed onto the surface, and the electrons are scattered in different ways depending on the contours. And that is how we get these 3D images, which we can see here in these different blood cells in a sample from the blood. You could be asked to calculate magnification of an image, so the formula that you need to remember is IM, and that is the image size equals actual size times magnification. Or you can rearrange it to work out the actual size or the magnification. And the key thing that you need to be aware of when you are doing these calculations is the image size and the actual size have to be in the same units. So you need to be familiar with how you convert from millimeters to micrometers or micrometers back into millimeters. Those are the two most common conversions in a magnification question. But if you want to know any of the other conversions, here they are. You might also need to be able to measure the size of a specimen using the eyepiece graticule. And inside of the optical microscope, there is a scale on a glass disc, which is called the eyepiece graticule. So it'd be just here within that eyepiece. And this can be used to measure the size of objects you're viewing under the microscope. However, each time you change the objective lens and therefore the magnification, you have to recalibrate the eyepiece to work out what the distance between each of these different divisions is actually worth. Now you need to know how to do that, but I'm not going to go through that in the summary video, but I'll link the video up here of how to calibrate the eyepiece graticule. Cell fractionation is the final way that you can study cells, and this is used to isolate the different organelles so that they can be studied further. First thing that happens is the cells are broken open to release the contents and the organelles. The cells need to be prepared in a cold, isotonic and buffered solution, and you need to know why it has to be those three conditions. So it has to be cold to reduce enzyme activity, and that's because when you break open the cell, you'll be releasing enzymes which wouldn't typically be in contact with the organelles and they might damage them. It has to be isotonic so that you don't have excess water moving in or out of the organelles by osmosis, because we don't want the organelles to burst or shrivel, because then we won't be able to study them. And it has to be buffered because if it became too acidic or alkaline, again, it could damage the organelles. Now, cell fractionation is a two-step process: homogenization and ultracentrifugation. So first of all, homogenization is when you break open the cells, and that can be done in a blender. As long as you have, as we said, the solution that you're blending up your sample in, as long as the solution that you're blending your sample in is cold, isotonic and buffered. Then we'd need to filter to remove the large debris, and then the filtrate, we can then use ultracentrifugation to isolate the different organelles. So we'd put our sample into a centrifuge and we'd spin at different speeds. The organelles are going to separate according to their densities, and that is why we have to spin our sample at different speeds. And this is what differential centrifugation is. So when we spin our sample, the centrifugal forces cause pellets to form at the bottom with the most dense organelles. So we can see here in this first image, we've got all of the organelles equally distributed. And if we first spin at low speed, the most dense organelle will form a pellet at the bottom. We would then remove that pellet and spin the rest of the filtrate again at a slightly faster speed. We'd then get the next most dense organelle forming the pellet, and we repeat this process at increasingly faster speeds. So each time that liquid, which is the supernatant, is removed, and the pellet is what the organelles will be in, and we can examine those. Now, just to let you know the order of the density, in that first centrifugation, the nuclei are the most dense, so those would be isolated in the first spin. The next most dense are the chloroplasts and the mitochondria, then the lysosomes, endoplasmic reticulum, and finally the ribosomes. So that would be the order in which you would collect your different organelles. We then move on to cell division, and eukaryotic cells enter the cell cycle and divide by mitosis or meiosis, but in topic 1 you only learn about mitosis. In comparison, prokaryotic cells replicate by binary fission, and viruses do not undergo cell division at all because they are non-living. They do still replicate though, but viruses replicate inside of a host cell. They will invade that host cell by injecting in their nucleic acid, so their genetic material, and then it will be the host cell that uses that genetic material to replicate the virus particle. The cell cycle that eukaryotic cells will be going through includes these key stages. Interphase is the longest stage of the cell cycle, and it includes G1, S, G2. G1 is when the cell is going to be increasing in size and the organelles will double. S phase is when DNA replication happens. G2, you'll have further growth, but also it says preparation for mitosis. In G2, you'll have this error check stage. So if there are any errors in the DNA replication, the cell would be destroyed at that stage. Nuclear division is either mitosis or meiosis, but in topic 2, we just focus on mitosis. The final stage of the cell cycle is cytokinesis, and this is when the cytoplasm divides to create the two new cells if it's mitosis. Mitosis is split into 4 key stages, which are our PMAT, prophase, metaphase, anaphase, and telophase. The key facts about mitosis are it's only one round of division, genetically identical cells are created, the cells are diploid, which means there are 2 copies of every chromosome, and this stage is used for growth and repair. So a specific example of that, which we'll see later in this video, is the clonal expansion of B cells. So that is growth in the sense of we're creating lots of new cells. So in prophase, the chromosomes will condense and at this point they become visible. And in the animal cells, the pairs of centrioles will move to opposite poles, which means opposite sides of the cells. The centrioles are going to be creating these spindle fibres, which are released from both poles to create the spindle apparatus. And these will attach to the centromere and the chromatids on the chromosomes in the later stages. Plants have a spindle apparatus, but they don't have the centrioles. In metaphase, we can see that the chromosomes will then line up in single file along the equator, and the spindle fibres are released from those centrioles at the poles, and they'll attach to the centromere and also the chromatids. In anaphase, the spindle fibres will start to retract and pull back towards the centrioles, and in doing that they'll pull on the centromere and chromatids, and this causes the centromere to divide in 2, and the individual chromatids are pulled to the opposite poles of the cell, and therefore it separates the chromatids, and once those chromatids are separated, we now actually call them chromosomes. Now this stage requires energy in the form of ATP, and that's provided by respiration from the mitochondria. In telophase, the chromosomes are now at each pole of the cell and become longer and thinner. The spindle fibres will then disintegrate and the nucleus starts to reform. The final stage in the cell cycle is when the cytoplasm splits in 2 to create the 2 new genetically identical cells, which the mitotic index can be calculated by counting first of all how many cells are visible in the field of view, and then counting the number of cells that are also visible but in a stage of mitosis. You can then do a percentage calculation. So you'd be doing the number of cells that you can see in a stage of mitosis divided by the total number of cells present and times it by 100. Prokaryotic cells don't go through mitosis, instead they go through binary fission. The first step of that would be the replication of the circular DNA and of the plasmids, and then the second stage would be division of the cytoplasm to produce 2 daughter cells, each with a single copy of the circular DNA and a variable number of plasmids. Viruses are non-living, so they don't undergo cell division. Instead, they inject their nucleic acid into the host, and the host cell replicates the virus particles. So now onto plasma membranes. All cells and organelle membranes have the same structure. It's this fluid mosaic model which is named because of the fact that it does have some slight movement, and also it's composed of a range of different molecules such as phospholipids, proteins, glycoproteins and glycolipids. All of these molecules are arranged within the phospholipid bilayer, and they create the partially permeable membrane. To create the bilayer, the phospholipids align in this manner because of the fact that the phospholipid has a hydrophilic head due to that negative charge on the phosphate group, and that causes it to be attracted to water. Whereas the hydrophobic tails, those are repelled by water but can interact with lipids. So the phospholipids end up spinning round so that the tails are opposite each other, the heads are on the outside where they can interact with water. Cholesterol is present in some membranes too, and this will restrict the lateral movement of other molecules in the membrane. This is useful as it makes the membrane less fluid at high temperatures and prevents water and dissolved ions from leaking out of the cell. The other components of the membrane are mainly the proteins, and these are embedded across the cell surface membrane, either as peripheral, meaning they're just on the outside, sometimes called extrinsic, or they could be integral, sometimes called intrinsic, meaning it spans from one side of the bilayer to the other. The peripheral proteins provide mechanical support, or they can be connected to proteins or lipids to make glycoproteins and glycolipids. The peripheral proteins provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and also carbohydrates can bind to the lipids directly to make glycolipids. The function of these is cell recognition or as receptors. The integral proteins are protein carriers or protein channels involved in the transport of molecules across the membrane. Protein channels are tubes, and these fill with water, which will enable water-soluble ions to diffuse across the membrane, whereas the carrier proteins will enable molecules to bind with them, and therefore larger molecules like glucose and amino acids can cause those carrier proteins to change shape and therefore transport the molecule to the other side of the membrane. So when we say that the membrane is partially permeable, what we mean by that is only lipid-soluble substances and very small molecules can pass across the membrane by simple diffusion. Other molecules, such as water-soluble or polar substances and large molecules, can't simply diffuse across the membrane. Therefore, they have to be transported across by other means, which could be facilitated diffusion, active transport, or if it's water, osmosis. So simple diffusion first of all, this is the net movement of molecules from an area of higher concentration to an area of lower concentration, and this will continue until an equilibrium is reached, and this process does not require any ATP. The molecules do still have energy to be able to move, but this is just due to the kinetic energy that they possess, and that is why they're able to move constantly as a fluid. For molecules to diffuse across the membrane, they have to be lipid-soluble and small. If they're not lipid-soluble or small, then it will be facilitated diffusion instead. And this is still a passive process because the molecules are moving from an area of higher concentration to lower, but because they can't simply diffuse through the phospholipids, instead they have to diffuse through proteins. Now this could be through protein channels, where we have those tubes filled with water, and therefore water-soluble ions can dissolve and then diffuse through. This is still selective though, as the channel proteins only open in the presence of certain ions when they bind to the protein. Carrier proteins will bind with a molecule such as glucose, which causes a change in the shape of the protein. This change enables the molecule to be released to the other side of the membrane. Osmosis is the movement of water, and that will be from an area of higher water potentials to an area of a lower water potential across a partially permeable membrane. The water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. Pure water or distilled water has a water potential of 0. So you cannot get one any higher than 0. It's always going to be 0 or negative. And that means when solutes are dissolved in water, the water potential will be negative. And the more negative the water potential, the more solutes there must be dissolved in it. Now, 3 key terms that you need to know linked to osmosis are isotonic, hypertonic, and hypertonic. An isotonic solution is when the water potential is the same in the solution and the cell. Hypotonic is when the water potential of the solution is more positive, so closer to 0 or closer to pure water compared to the cell. Hypertonic is when the water potential of the solution is more negative than the cell, so there'd be more solutes dissolved in it. And in an animal cell, we can see the impact that would have. In an isotonic solution, because the water potential is the same, there's not going to be any net gain or loss of water. In a hypertonic solution, there'll be more water moving out of the cells by osmosis, and that can cause animal cells to shrivel or crenate. If animal cells, such as red blood cells, are put into a hypotonic solution, that would mean water would be moving into the cell by osmosis, and if enough water moves in, it can cause the cell to burst. Active transport is the movement of substances from a lower concentration to a higher concentration, and this requires metabolic energy and a carrier protein. So the process by which this happens is, first of all, it's always through carrier proteins only. It is not channel proteins for active transport. The molecule will bind to the carrier protein, and it can only bind to proteins which have a complementary receptor shape, so it is still selective. The ATP binds to the carrier protein as well from the inside of the cell, and it is hydrolyzed into ADP and PI. This causes the carrier protein to change shape, and therefore it releases the molecule to the other side of the membrane. The phosphate ion is then released, and the protein can return to its original shape, and the process can then happen again as long as there is plenty of ATP present. Co-transport is a type of active transport, and you learn about it across the course, but one of the key examples you learn about is the co-transport of glucose, or it could be amino acid, with sodium ions in the ileum. And to absorb glucose from the lumen of the gut, there has to be a higher concentration of glucose in the lumen compared to the epithelial cell. But there's usually more glucose in the epithelial cells, and that is why active transport and co-transport are required. So the first thing that has to happen is the sodium ions, which are shown in blue here, are actively transported out of the epithelial cell into the capillaries. And that will result in a lower concentration of sodium ions within the epithelial cells. And that creates this concentration gradient from the ileum to the epithelial cell. And therefore the sodium ions can diffuse down their concentration gradient into the epithelial cells. The proteins the sodium ions diffuse through, though, is a co-transporter protein. So either glucose or amino acids will also attach and are transported into the epithelial cell with the sodium ions, except the glucose is going in against its concentration gradient. The glucose, once in the epithelial cell, can then move by facilitated diffusion from the epithelial cell into the blood. Now, you also need to be aware that cells may be adapted for rapid transport across their internal or external membranes. So it could be that cells have an increased surface area, so things like microvilli, for example. Or there could be an increase in the number of protein channels and carrier molecules within the membrane as another way to increase the rate of transport. The last section of topic 2 is all about immunity, and we begin by looking at identifying self and non-self cells. So your body's immune system has cells to be able to identify the presence of pathogens and potential harmful foreign substances compared to your own body cells. And that's so it can destroy the foreign items and not your own cells. And those cells are called lymphocytes. But how can lymphocytes then actually distinguish between a pathogen and your own body cells? Well, each type of cell has specific molecules on its surface that identify it. And these molecules are usually proteins, so they'll have this unique 3D tertiary structure. And this is how they can identify whether it is an antigen from a pathogen or whether it is one of your self-antigens. If a non-self cell is detected, a response will be triggered to destroy that cell. And these different surface molecules enable them to identify pathogens, which we've mentioned, so bacteria, fungi, viruses, but it could also be cells from other organisms of the same species, and that can be harmful for individuals who have had organ transplants. It could also be abnormal body cells, for example, cancer cells, and finally toxins, because some pathogens release toxins into the blood, such as the bacteria cholera. Now, an antigen is a molecule that generates an immune response, and this is because it triggers the lymphocytes. They are usually proteins and they are located on the surface of the cells. Now, antigen variability is this concept that pathogens' DNA can mutate frequently. If a mutation occurs in the gene which codes for the antigen, then that means the shape of the antigen on the outside of the virus, the fungus, the bacteria will change. And if that happens, any previous immunity that you may have had to the pathogen is going to be lost. So it will no longer be effective, as all of the memory cells in the blood that you have will have a memory of the old antigen shape and not the new one. And this is known as antigen variability. The influenza virus mutates and changes its antigens very quickly, and this is why a new flu vaccine is created every year to account for that change. So looking at your immune response then, if a pathogen gets past the chemical and physical barriers, so for example your skin is a physical barrier and stomach acid is a chemical barrier, then your white blood cells are the next line of defense. Your white blood cells have a specific and also non-specific response. The phagocytes are a non-specific response, meaning that they will destroy any foreign item that they come across. Lymphocytes have a specific response though, and this is where the idea of detecting using antigens will come into it. So let's look at phagocytosis first of all. A phagocyte is a macrophage cell, which is a type of white blood cell, and they are found in the blood but also tissues. Phagocytosis, as we said, is a non-specific response. So any non-self cell that is detected will trigger the same response every time to cause the destruction of that object. So the stages of phagocytosis are, first of all, the phagocytes are in the blood and the tissues, and any chemicals or debris released by the pathogens or abnormal cells attract the phagocytes, and that will cause those white blood cells to move towards the cell. There are many receptor binding points on the surface of the phagocyte, which we can see here. They will attach to the chemicals or the antigen on the pathogen via the receptors. The phagocyte then changes shape to move around the pathogen and engulf it, and once engulfed, the pathogen is contained within a vesicle, which we call a phagosome. A lysosome within the phagocyte will then fuse with the phagosome and it will release its contents. And the contents is a lytic enzyme called lysozyme. And lytic enzymes are able to hydrolyze. So the result is that that enzyme will hydrolyze and it breaks down the pathogen. Any soluble products are absorbed and used by the phagocyte, and anything else is released as waste and debris. So that is our non-specific response, but now if we look at the specific responses, which are by the lymphocytes, first lymphocyte we're going to look at are the T lymphocytes, or the T cells. So all lymphocytes are made in the bone marrow, but the T cells mature in the thymus, which is why they're called T cells. And this is classed as the cell-mediated response. So the first thing to be aware of is the cell-mediated response is specific to T cells responding to antigens on the surface of cells. So it involves antigen-presenting cells, or APC. An antigen-presenting cell is any cell that presents a non-self antigen on their surface. Infected body cells will present the viral antigens on their surface if they are infected. A macrophage, which has engulfed and destroyed a pathogen, will also present the antigens on their surface. Cells of a transplanted organ will have different shaped antigens on their surface compared to your own self cells. And cancer cells will have abnormal shaped self antigens as well. So all of these are presenting antigens on the cells, so they are antigen presenting cells and can therefore trigger an immune response. So the cell mediated response then, once a pathogen has been engulfed and destroyed by a phagocyte, the antigens are positioned on the cell surface. And that is why we'd now call that phagocyte an antigen presenting cell. Helper T cells, which are a type of T lymphocytes, have receptors on their surface that can attach and bind to the antigen on antigen-presenting cells. And once those two cells are bound, it activates the helper T cell to start to divide by mitosis. And that then means we get a very large number of cloned cells. So essentially, this stage is to make sure you end up with lots of helper T cells which have the correct shape to bind on to that particular antigen. So you'll have lots of helper T cells within your blood that have different shape receptors to be able to bind onto different shaped antigens. But if you have one collide, that will then activate that particular T cell with that particular shape receptor to divide by mitosis to make large cloned copies. Those cloned helper T cells then differentiate into particular types of T cells. Some remain as helper T cells, which then go on to activate the B lymphocytes. Some can stimulate macrophages to perform more phagocytosis. Some become memory cells for that particular shaped antigen. And some become cytotoxic killer cells or cytotoxic T cells. And what those cytotoxic T cells do is destroy the abnormal or infected cells that have the antigen on their surface. And they do this by releasing a protein called perforin, which embeds in the cell surface membrane to make a hole or a pore, so that any substance can now enter or leave the cell. Now, this causes death because you can have lots of water move in and therefore the cell will burst, or you could have lots of water moving out and therefore the cell will actually shrivel up and die. This is most common in viral infections because viruses infect body cells of the host. Body cells therefore have to be sacrificed to prevent further viral replication. And this is actually why you get a really sore throat when you have a cold, because those cytotoxic T cells are destroying the infected body cells in your throat to try and prevent that virus dividing and spreading further in your body. Next then, we move on to the B lymphocytes or the B cells. As we already said, all lymphocytes are made in the bone marrow, but B cells mature in the bone marrow as well, which is why they're called B cells. This is now called the humoral response, and this is the one that involves antibodies. Antibodies are soluble and transport in bodily fluids, and humour is an old term for body fluids, and that's why this is called the humoral response. There are approximately 10 million different B cells which have antibodies on their surface, complementary to 10 million different antigens. And antigens in the blood will collide with their complementary antibody on a B cell, and the B cell takes in the antigen by endocytosis and then presents it on its cell surface membrane. When that B cell collides with a helper T cell, that can then activate the B cell to go through clonal expansion and differentiation, or clonal selection. So this is the step where there's that link between the cell-mediated response with the helper T cells binding via the receptor to the antigens on the B cells to activate them. Once activated, the B cells undergo mitosis to make large numbers of the cells which have that particular antigen on their outside. And these can then differentiate into plasma cells or memory B cells. The plasma cells will go on to make antibodies complementary in shape to that particular antigen. And the memory cells can divide rapidly into plasma cells if you are reinfected with the same pathogen and therefore antigen later on. And that means that you can make large numbers of the correct shaped antibody so rapidly that you should be able to destroy the pathogen before it causes any damage and symptoms. So the memory B cells can live for decades in your body, whereas the plasma cells are only short-lived. Memory B cells, though, cannot make antibodies, but they can divide by mitosis and then differentiate into plasma cells. So this is what results in the large numbers of antibodies being produced so rapidly that the pathogen is destroyed before any symptoms occur. And that's what it means when we say you are immune to a particular disease. It's because you have these memory B cells, so you can destroy the pathogen before it causes any symptoms. And that would be an example of active immunity. Primary and secondary response is referring to the number and the speed at which antibodies are produced when you are first exposed to an antigen compared to your second exposure. So the first time you're exposed to a new pathogen and therefore a new antigen, it takes a little bit longer for the antibodies to be produced because you have to go through that initial step of those 10 million different B cells colliding until you have the correct shape antigen-antibody binding. So that can take a couple of days, and that's why you have a slower response in producing the antibodies, and also you don't actually make as many. But because memory B cells are made, if you are exposed for a second time, you will be able to create those antibodies very, very rapidly and in much larger numbers. And this is what is meant by the primary response and the secondary response. So if we look a bit more at antibodies, antibodies are an example of quaternary structured proteins because they're made up of 4 polypeptide chains. The part shown in purple is described as the variable region, and that is where you would have the part of your antibody that binds onto particular shaped antigens. The part in red is constant, so it would be the same for different antibodies. We can then see we have one longer chain, which is heavier, so we call it the heavy chain, and we have a shorter chain, which is called the light chain. And this bit here where it is variable, as we said, that is the antigen binding site. Now those antibodies are actually flexible, and that enables an antibody to bind to multiple antigens. So we can see we've got binding to 2 antigens here, and then this antibody is binding to 2 as well, and as a result they all end up clumping together, and we call this agglutination. The advantage of agglutination is if you've got a big clump of antibodies and antigens which are going to be attached to the pathogen, it makes it much easier for the phagocytes to locate and therefore destroy the pathogens more rapidly. So we've talked about active immunity. Passive immunity, though, is when the antibodies are introduced to the body. So you haven't made the antibodies yourself, you are just gaining the antibodies. So that means that the pathogen didn't cause the creation and differentiation of plasma cells or any memory cells, so you won't have any long-term immunity. Examples of passive immunity could be antibodies passed to a fetus through the placenta, or through breast milk from the mother to their baby. Active immunity, as we said, is when the immunity is created by your own immune system following the exposure to the pathogen. But this can be either natural or artificial. And natural is following an infection where you've actually had that pathogen within your body. Artificial is following the introduction of a weakened version of the pathogen or antigens via a vaccine. So if we go through then how vaccines work. Small amounts of weakened or dead pathogen, or it could just be the antigens, are introduced either to the mouth or injected into the body. An exposure to those antigens activates the B cells to go through clonal expansion and differentiation. So that was the clonal selection part where we have mitosis occurring to make large numbers of those B cells. Those then differentiate into plasma cells and memory B cells. As we said earlier, the plasma cells will make antibodies, but the key bit of importance for a vaccine is the fact that B memory cells will also be produced, and those can stay in the blood for years. So that means that if you are infected with the actual pathogen, those memory B cells, if they collide with the antigen, will divide rapidly into plasma cells when you are reinfected, and therefore you create large numbers of antibodies so rapidly that you shouldn't get the symptoms of the disease. Or if you do, they'll be very, very mild and you should be able to overcome the disease more quickly. Now, not everyone is able to take vaccines for various reasons, but the concept of herd immunity is if enough of the population are vaccinated, the pathogen cannot spread easily amongst the population anymore. So this provides protection for those who aren't vaccinated. For example, if you already have another illness, which means it'd be too dangerous for you to have the vaccine, if you have a lowered immunity, if you're too young, and sometimes if you're pregnant, you're not able to take vaccines as well. Now, the structure of HIV is made up of 4 key components. We have the core, and that is the genetic material, which is RNA, and the enzyme reverse transcriptase. And this is needed for the viral replication when it's in the host. You also have a capsid, which is an outer protein, which you can see just here. You then have the envelope, which is an extra outer layer made out of the membrane taken from the host cell membrane. And then we have protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cells. So the way that HIV replicates in helper T cells is that first of all, it's transported in the blood until it attaches onto a CD4 protein, which is one of the attachment proteins on the outside of helper T cells, which we can see just here. The HIV protein capsule then fuses with the helper T cell membrane, enabling that RNA and enzyme to be injected or enter into the host's cell. The HIV enzyme reverse transcriptase copies the viral RNA that's been injected into a DNA copy and moves to the helper T cell nucleus, and this is why it's called a retrovirus. Here, the mRNA is transcribed and the helper T cell starts to create viral proteins to make the new particles. Someone's described as HIV positive when they are infected with HIV. AIDS is when the replicating viruses in the helper T cells interfere with the normal functioning of the immune system. So someone who is HIV positive doesn't necessarily have AIDS. With the helper T cells being destroyed by the virus, the host is unable to produce an adequate immune response to other pathogens and is left vulnerable to infections and cancer. It is the destruction of the immune system that leads to death rather than the HIV directly. So the very final thing is this concept of monoclonal antibodies. Mono meaning one and clonal meaning identical. Antibodies are proteins, which we looked at, and they have these binding sites complementary in shape to a particular antigen. And this has been manipulated to create monoclonal antibodies. And that can be for medical treatments, medical diagnosis, it's used in pregnancy tests, it's also used in tests such as drug testing and testing for other viral diseases. So targeted medication, we're going to go through that example first. This is an example of direct monoclonal antibody therapy. So what we mean by that is some cancer can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of the cancer cells. The antibodies are given to the cancer patient and they will then attach onto the cancer cells only. While the antibodies are bound to the cancer cells, this prevents chemicals binding to the cancer cell, which will enable uncontrolled cell division. Therefore, the binding of these monoclonal antibodies prevent the cancer cells from growing. And as they're designed to only attach to the cancer cells, they're not going to harm any other normal cells. Indirect monoclonal antibody therapy can also be used to treat some cancers. The monoclonal antibody is created exactly the same, except there will be a drug attached to them. Indirect monoclonal antibody therapy starts with the same process of creating a monoclonal antibody which has a binding site complementary in shape to the antigens on the outside of cancer cells. but this time a drug is also attached to the antibody. The cancer drugs are therefore delivered directly to the cancer cells and can destroy those cells only. And this reduces the harmful side effects that traditional chemotherapy and radiotherapy can produce. And this is sometimes called a bullet drug because it's being directed to just one particular site. Medical diagnosis is another use, and as we said, this could be in pregnancy tests, viral tests, so influenza, could be in bacterial tests as well, cancer tests. Most recently, we've seen in COVID-19 lateral flow tests as well. And these use the ELISA or the ELISA test. So the ELISA test, which stands for Enzyme-Linked Immunosorbent Assay, or ELISA, uses two antibodies. So first of all, the first mobile antibody, which is complementary to the antigen being tested for and has a coloured dye attached to it, we can see just here. Part C, we have a second antibody, which is complementary in shape to the antigen as well, but this one is immobile, so it's attached to this fixed position. There is then a third antibody, which is immobilized at point D, and it is complementary in shape to the first antibody. So what this means is, if you are pregnant, if we think about this in terms of a pregnancy test, you contain the hormone HCG within your urine. That will then bind to the antibody, and any antibodies with the HCG hormone bound to it will then attach to the immobilized antigen at point C. And therefore, you get a coloured blue line at this point because we have that blue bead held in that fixed position. You should always have a second blue line, whether you're pregnant or not, at position D though, because this second immobilized antibody is just complementary to the shape of the first one. So it's basically just a control to check that you do definitely have that first antibody in your sample and it is able to move along the test kit. Now another test we can look at is this one here for the ELISA test, where you would add the test sample from a patient to the base of a beaker, wash to remove any unbound test sample. Then we add an antibody complementary in shape to the antigen you are testing the presence of in the test sample. Again, we then wash to remove any unbound antibody. Add a second antibody that is complementary in shape to the first antibody and binds to the first. The second antibody we can see here has an enzyme attached to it. Again, we have to rinse just to make sure if there's any unbound antibody, those are taken off. And then the substrate for the enzyme, which is colourless, is added. And this substrate produces a coloured product when that enzyme is present. So the presence of colour indicates the presence of the antigen in the test sample, and the intensity of the colour indicates the quantity present. Now there are some ethical issues surrounding monoclonal antibodies, and that is because within the creation of monoclonal antibodies, you do require animals such as mice to produce the antibodies and tumour cells. And that can lead to ethical debates as to whether the use of animals is justified to enable the better treatment of cancer in humans and to detect diseases, because this will cause discomfort, harm and death to the animals that are being used. So that is it for topic 2. If you do want to see any of it in more detail, because there are some quite complex topics there, then have a look at this topic 2 playlist. If you have found this helpful, please give this video a thumbs up.